Hello, my name is Karl Johan Westrin and I'm a PhD student in Olof Emanuelsson's group at KTH. Today and on my poster I will describe one of our ongoing projects about cluster-guided de novo isoform assembly. Here you can see a flow chart of this very pipeline. But before going into how it works, why should we look into this problem? Well, different isoforms are an important part when studying transcriptomes and alternative splicing in particular. But one of those things or problems with de novo assembly is that it requires deeper sequencing than ab initio or reference-based assembly. So when lacking a complete reference genome, one might miss some lowly expressed isoforms. So that's why we're looking into this. This flowchart might be hard for you to make out. I will show you an illustration on how the pipeline works. In the first step, we'll see the input, the short pad end reads, and an optional set of guiding contigs. It might be a set of long reads, a previous assembly, or a reference transcriptome. Then, in the second step, the short reads are assembled by Transibis, a tried and tested assembler, into a primary assembly, just in order to be able to catch up such genes that the later cluster-wide assembly might miss. Then the third step begins by de novo cluster the guiding contigs if such were given. If such weren't given, it will cluster the primary assembly from step 2 instead. By aligning the short reads to the guiding contigs, the short reads are clustered likewise. In the following step, each cluster is assembled individually with Shannon, and finally, in the last step, merged with one another into and the primary assembly, and that's the pipeline. We ran our pipeline called clustrust and 9 other de novo transcriptome assemblers on datasets from human, from mouse, from rice, from Arabidopsis, from zebrafish, and poplar. To show the benefit of the clustering step, we also included the results from just concatenating Transibis and Shannon whenever possible, because Shannon didn't finish on the rice dataset. Here we can see the results when calculated with Scanti. To make this easier for you to see, I will show you each species individually with the name of the current species in red. Feel free to pause the video to be able to look more carefully. So that was the result from Scanti. Now we will also see the same results when calculated with CRBB as well, and for each individual species. So, as we can see, clustrust recovered more expressed known isoforms than any of the other tested de novo assemblers, but with a moderate reduction in precision. Therefore, we propose that clustrust can be a useful tool when studying alternative splicing in non-model organisms. Thank you for your attention.